Hi, everybody. I would like to first thank the organizers for giving me the opportunity to attend and present at this really nice conference. I really enjoyed all of the talks, and that discussion was so frank and passionate, and I really feel very hopeful. So I'm at the Institute of Molecular and Cell Biology, and the main biological question that my lab is trying to understand and hopefully contribute to solving is very broadly neurodegenerative diseases. So as you know, with our aging societies, neurodegenerative diseases whose prevalence increases with increased age are getting more and more prevalent, as well as having great societal and medical costs. So for example, just looking at two of the most common neurodegenerative diseases, Parkinson's disease as well as Alzheimer's disease, the worldwide prevalence is up to 10% of the population over 65 years. So obviously, it's a very important question, and many pharmaceutical companies and academic institutions have spent billions of dollars on clinical trials, many clinical trials, many drugs, but to date, there's still no disease-curing drugs for these diseases. And we're coming to realize that we really need to understand the mechanisms that underlie these diseases, because a lot of the drugs have been focused on trying to clear the plaques that we see in the brains without really understanding why the plaques are there, for example. And so in my lab, we actually use the fly model to study this problem. And some people, my mom used to ask me, like, does the fly even have a brain? Well, the fly does have a brain, and despite being a very small animal, it actually needs to carry out a lot of very complex behaviors. So the fly can learn, it courts, it can look for food, and obviously the fly can fly, which we cannot do. And it does this with a brain that is many orders of magnitude smaller than human brains. So in terms of number of neurons and total number of synapses, the fly brain is 1,000,000-fold less complex. So you can imagine that if you're trying to understand a problem and you can distill it down to 1,000,000-fold less complexity. Obviously, this is a simplified idea, right? So rather than studying a million neurons, you could study one neuron. Maybe we could really gain insight into fundamental aspects of the problem. And importantly for us, 2/3 of disease-causing human genes are estimated to be present in the fly, which is important because we want to study the disease. And we can study and manipulate processes in situ with very powerful tools in the fly. So just as an example here, I want to show you that in the fly, we have tools that allow us to control and understand the function of individual neurons. So here on the left, you see a normal fly walking. And on the right is when this group has activated a group of 7 neurons in the fly. And when you do this, the fly starts to walk backwards. So with these very powerful tools, we can actually understand at the single neuron level what every single neuron is doing, and then how they work in circuitry to generate normal behavior, as well as when these circuits are lost, what happens in disease. So more specifically, my lab is interested in movement disorders. So if we think very broadly, neurodegeneration typically can result in 2 different types of phenotypes, either cognitive or movement. Obviously, there's also diseases that have both phenotypes. So movement disorders are neurological conditions that cause involuntary abnormal movements. So very broadly, they may either reduce your ability to create the movement that you want, or you may have excessive movements. So the first category are called hypokinetic movement disorders, so less hypo. For example, Parkinson's disease. And the latter category are hyperkinetics. You see excessive movement. You create more movement than you want. So I'm going to show you 2 videos now of a doctor demonstrating these 2 types of disorders. So for example, in Parkinson's disease, it's associated with hypokinetic. It's a hypokinetic disorder. You have inability in, I didn't want the volume. Okay, so basically it's associated with rigidity. So you want to make a normal walking movement, but you're unable to make that movement. In fact, you're making very small shuffling movements. On the other hand, spinal cerebellar ataxia is associated with excessive movement. So you start to generate these jerky, lurching movements. So rather than making very smooth walking movements, you generate excessive movements. So in the clinic, these gait and tremor symptoms really are a clue for you to know which part of your brain is malfunctioning and which circuits have disorder. And they're important for clinical diagnosis, but also very important for us to be able to understand why different disorders result in different types of movement disorder, and giving us a clue as to which are the neurons that we need to understand to fix. So we started this project with a very simple question, which is, do these very simple animal models of neurodegeneration, for example, for Parkinson's disease, PD, and spinal cerebellar ataxia, SCAR3, do they walk differently from each other? And you can imagine that obviously the fly and the human are very different, and when we started this project, we don't know what the answer is. So we first wanted to ask, can we actually use the fly to distill the problem of movement disorders down to a simple animal by asking what do their gaits look like first? So to do this, we first needed to develop a way to study these very tiny movements in the fruit fly. So in collaboration with a machine learning lab, we developed a machine learning program to automatically track leg movements in wild-type flies as well as disease flies. And I won't go into the details of the program, but the unique feature of our method is that it doesn't require you to manually annotate training samples, which you usually need to do for machine learning. So normally, for example, if you wanted to train a machine learning program to learn dog from wolf, you need someone to tell them this is dog, this is dog, this is wolf, and then you train the program. But we developed a way to be able to do it without this manual annotation. This program allowed us to be able to look at fly walking. So we put the fly into an arena, and then the machine will automatically track the movements of the fly. So you can see here that a wild-type fly walks in a very smooth manner. The legs show very deliberate movements, that the footprints are very regular. The domains that each leg covers does not overlap with each other, and the domains are quite equal in size. So this is what a wild-type fly looks like. So with this method, we can now ask the question of whether the fly models of SCAR3 walk differently from the fly models for PD. And these fly models have been in study for about 2 decades. So for SCAR3, which is a disease caused by a single gene, we had 1 genetic model. And for PD, which the familial forms of the disease can be caused by different genetic defects, we used 2 different fly models. So we we know what these diseases look like in human patients. So for example, as I showed you in the video before, SCAR3 is associated with veering movements, erratic foot placement. You're not always having a very deliberate footprint. You have these lurching steps, while PD is not associated with these veering, lurching movements, but instead exhibit leg rigidity as well as shuffling. So we went in to look at these different fly models as well as control flies to look at what their walking looks like. So again, let me remind you that control flies walk in a very smooth, regular manner. So you can see the footprints of the flies fall in about the same spot, and the leg domains do not overlap. Now SCAR3 flies, so SCAR3, if I can remind you, is a hyperkinetic disorder. You saw the gait of the human patients typically having exaggerated big movements, and they are not able to have the regular footfalls, and their legs will frequently cross over with each other. So you can see here that when we looked at the SCAR3 flies, they no longer showed the very smooth movements of the legs, and the leg domains traversed by each leg could tend to overlap with one another. They also showed action tremors, and the domains of each leg covered a much larger area than would be seen in a normal wild-type fly. On the other hand, when we looked at a PD fly, you can see here that it doesn't look like a wild-type fly in that the movements are much slower, but in particular, the hind leg domains, if you can see, tend to be much smaller, showing a bit of stiffness. So you can see that in the domain of the distance traversed by the leg, which are shown in these colored leg traces, you can compare the hind leg traces of the PD flies, and they're much smaller than that of the control flies. So this reflects a type of rigidity because the leg is not able to traverse the normal leg stride length. So having these videos are nice, but we want to be able to measure these different parameters that we spoke about. So in order to do that, because these parameters lie on different axes, right? So if I were to just show you each parameter, all you'll see is a mess of plots. So we wanted to be able to develop a gait signature for each of these different disease models. And to do this, we made use of a statistical index called Cliff's Delta with our collaborators. And what Cliff's Delta does is allow us to plot each of the gait parameters that we're interested in. So for example, here on the y-axis, you see body veering, footprint regularity. So all of the gait parameters are now being able to present on the same plot. And what Cliff's delta does is, a Cliff's delta of 1 means that all of the mutant points are higher than the control points, while a Cliff's delta of -1 means that all of the mutant points are below that of the wild type in terms of numerical value, whilst a Cliff's delta of 0 means there's no difference between wild type and mutant. So you can see here that by eye, plotting at the Cliff's delta, the SCA3 flies and the PD flies show a very different gait signature. And as I mentioned, we had a separate, different genetic model for PD that was unrelated to the first model. And when we looked at these flies, they showed a very similar rigid gait signature compared to the first genotype. So 2 different unrelated fly models of PD, for which there's a perturbant, different genes are perturbed, show a very similar gait signature that differs from the SCA3 signature and exhibits a rigid phenotype associated with PD. So we wondered whether maybe the PD signature is just a signature that is seen in any fly that has some sort of degeneration because they're walking slowly. So then we looked at a fly model of glial degeneration, and you can see that the glial degeneration model does not show the hind leg rigidity phenotypes that is observed in the PD flies. And then we wondered whether we could understand why the PD flies show this very specific gait signature. And we asked whether if you only perturb dopaminergic neurons, which are the neurons that preferentially degenerate in human patients with PD, and has been shown to preferentially degenerate as well in PD fly models. So if we were to express the mutant SCA3 protein, which is causing the ataxic fly phenotype, if we now put them only in dopaminergic neurons, we saw that this recapitulated the rigidity phenotype. So this showed us that the rigid phenotype that's observed in flies really comes from dopaminergic neuron dysfunction, similar as that in humans. So now what we're doing is we are using our FLIT, our machine learning method, to study tremor, which is associated with many neurodegenerative diseases. And it's very common, but its causes are not well understood. It also has a very high genetic heritable risk. And we think that the reason that we know very little about the genetic causes of essential tremor, and as well as the mechanistic circuitry that leads to tremor, is because there's been no really good animal model. So we can now carry out automated leg tremor measurement, and we are using our machine learning method to automatically measure tremor by perturbing different populations of neurons to be able to understand the circuitry that leads to tremor, as well as what's going on in those specific neurons that causes tremor. Okay, so in summary, we developed a fully automated machine learning method for accurate leg tracking and gait analysis. And we, for the first time, realized that fly models for PD and SCA3 actually walk differently, just like in human patients. And genetically unrelated PD flies exhibit similar gaits. And we are able to now use the fly model to study tremor, as well as circuits and mechanisms. So I'd like to acknowledge the people in my lab who did the work, particularly Animesh, Kajun, and Mumita. And I was asked to talk a little bit about my experience as a woman in science and what sort of advice I would give. So I think number one, and something that I always say, is that actually scientists we are just really normal people. We are also flawed, we are not geniuses. So I think that this holds women back, because we think that, okay, to do something, we need to fulfill 80%, 90% of the criteria. But really, and then we build the criteria up very high in our head. But really, like, if you are curious, you have really good attention to detail, you're very rigorous, and you're very persistent, I would say, like, these are the traits that make a good scientist. And number two, find good mentors. And I think they talked a little about this in the discussion group, right? Sponsors or people who really care about your career progression. And those are the people who I've been very lucky to have very good mentors in my career. And lastly, this again something that they touched upon in the discussion group is to develop a good support network and people who will celebrate your successes and also cry with you when you fail, because there's a lot of failure. And if you don't, that support network is really what will carry you along. And I'm glad to meet many people here at this conference, and I hope that we can keep in touch and be that support network for one another. So thank you very much.